Hello everyone, today we will be going over staining microscopic specimens. Throughout this presentation, I will discuss why microscopic specimens are stained, the differences between simple and differential staining, the types of staining techniques, and the procedures and applications for each of the techniques. So first of all, why do we even stain microscopic specimens? Well, staining specimens gives them color and contrast that make them more visible under the microscope, which also allows us to detect their cell morphology, such as their shape and the way that they are arranged. Staining will also allow us to identify unknown microorganisms based on the results of each stain. Now, the main difference between simple and differential staining is how many dyes are used and what the stains achieve. Simple staining uses only one dye that either stains the cell or the background, which is going to enable their structures and characteristics to really pop out. If just the cell itself is stained, then they will all be the same color regardless of their species. There are two different types of simple staining techniques, which are simple and negative stain. For differential staining, many dyes are used, which will allow us to distinguish the organisms based on the reaction of the stains. Since there are so many stains that can be used, depending on the procedure, all stains must be used in the correct order or you will not get accurate results. There are 5 different types of differential staining techniques, which include gram stain, acid-fast stain, capsule stain, endospore stain, and flagella stain. For our first simple staining technique, this stain, the simple stain, is a positively charged basic stain that stains the negatively charged molecules and structures of the organism. So long story short, this stain will paint the cell, making the cell's morphology more visible. To complete this stain, you must first heat fix your bacterial smear. Heat fixing is where we apply the slide to a heating element for a short amount of time to attach the specimen to the slide to ensure that it does not move and that it doesn't continue to grow. The primary stain is applied to the slide, completely covering the specimen, which is going to color all of the cells. And a primary stain that can be used is either safranin or crystal violet. Here you can see that the cells have been stained like a purple pinkish color and that the background remains unstained. And then you can also notice that it has a streptobacilli morphology, meaning that it is in a chain and that the cells are rod-shaped. The negative stain is another simple staining technique which uses a negatively charged acidic stain which stains the background and leaves the cell to appear to be clear or white, and it once again allows us to visualize the cell's morphology. To complete this stain technique, you will add acidic dye to the slide, submerge a few cells of the specimen into the dye, and then spread the mixture from one side of the slide to the other. And then once we do that, we won't heat fix this, but we will allow it to air dry. And now, depending on the acidic dye that you use, here, the stain that was used is India ink, which will stain the background black, but you will still see that your cells are going to appear white or clear, showing that this specific bacteria has a diplobacilli morphology, meaning that it's rod-shaped and paired. For our first differential stain, the gram stain, it's a differential technique that distinguishes bacteria based on their cell wall structure. This stain is also used to classify bacterial pathogens. So here you can see that the gram positives end up being purple after the procedure is complete. It's because crystal violet, the primary stain, whenever it is added, it will bind to the peptidoglycan that is within the gram-positive cell wall. And then here for gram-negative, you can see that it will result in a pink color because the counterstained safranin will dye the cell pink because Gram-negative bacteria contain lipopolysaccharides in its cell wall, which the primary stain crystal violet does not bind to. For this technique, we will prepare a bacterial smear and then flood the slide with the primary stain crystal violet to the cells. Next, the mordant, which is Graham's iodine, will be added to the slide. The mordant pretty much just increases the binding of the primary stain to the cells if the peptidoglycan is present in the cell wall. Next, you will decolorize your cells, adding 95% ethanol. The decolorizer will remove any stain that did not bind to the specimen cell wall. Finally, you will add the counterstain, which will stain the cells that were decolorized or did not retain the primary stain. So here you can see the step-by-step procedure for both gram-positive and gram-negative. And you can see that for both of them, when the crystal violet was added, they will actually both be stained the purple color. And then the Graham's iodine will come in and treat the primary stain to get it to really stick to that cell wall. But once we get to the decolorization stage where the 95% ethanol is added, the gram-positive will actually retain the crystal violet once again because it contains that peptidoglycan, while gram-negative will actually lose the crystal violet because it does not contain the peptidoglycan. Instead, it contains the lipopolysaccharides, which will then be counterstained with the safranin. The acid-fast stain differentiates gram-positive organisms that have mycolic acid in their cell walls and those that do not. This technique is used as a diagnostic tool to visualize resistant organisms. The primary stain actually stains the cell wall of organisms that contain mycolic acid, which is a waxy coat that protects bacteria from drying out and even protects them from some antibiotics. This technique is also used to test for Mycobacterium tuberculosis, which is the main bacteria that causes tuberculosis. There are two methods of acid-fast staining. The Ziehl-Neelsen method, which uses heat as a mordant to infuse the primary stain into the cells, whereas the Kenyon method does not use heat as a mordant. The procedure for the Ziehl-Neelsen method is to add the primary stain carbofuscin to the slide while it's on the heat source. And then the heat as the mordant will help the primary stain bind to the layer of mycolic acid. And then the decolorizing agent, which in this case is acid alcohol, will be added to the slide to remove any excess primary stain from the slide. And then you will place the slide back on the heating element and add the counterstain, methylene blue or brilliant green, to stain the cells that did not retain the carbofuscin. So here in the picture, you can see that here this cell, all these pink cells, retain the primary stain carbofuscin because those cells actually contain the mycolic acid layer, and so those would be your acid-fast cells. And then these blue cells are going to be your non-acid-fast cells that retained the methylene blue because they do not contain the mycolic acid, which are going to be your non-acid-fast cells. Capsule staining is a technique that stains around a bacterial capsule with an acidic dye and then the cell itself with a basic dye. And whenever that's done, it's going to leave the capsule, if present, unstained, which kind of makes it look like a halo around the cell. This stain allows us to differentiate organisms that contain capsules from those that do not. To conduct the capsule stain, acidic dye is added to the slide, and then the specimen is emulsified into the dye, which is then spread on the slide from one side to the next, and it will stain the background around the cell. Once the acidic dye has air dried, a basic dye is added to the slide, flooding the cells and staining them. So if you look here, you can notice that the background of this slide is kind of like a pinkish purplish color. And then these cells present are going to have a purple, more dark purple color. And then here you have this white area. That is actually going to be your capsule that did not get stained. So these two are going to be your two cells that have capsules. and then these two cells are going to be the cells that do not have capsules. Endospore staining is a differential staining technique that uses two stains to distinguish organisms with endospores from those without. The endospores will appear a bluish green, while other structures will appear pink or a pinkish red. Well, first of all, let me explain what an endospore is. An endospore is kind of like a dormant structure within the cell that functions as a survival mechanism when environmental factors such as heat or light or oxygen are not the best for the cell. So if the temperature is too high or low, it could actually cause damage to the cell, potentially killing the cell. But these endospores, they contain their genetic structures and actually protect the cell. This staining technique is used to identify Bacillus and Clostridium bacteria, which is very important because Bacillus anthracis is a bacterial species that causes anthrax, and Clostridium difficile is the bacterial species that is responsible for the hospital-acquired infection, which is also known as C. diff. This technique is also used to further study endospores. To conduct this stain, the primary stain, malachite green, is flooded onto the stain, covering the heat-fixed specimen. Heat in this procedure, heat is going to be the mordant. So whenever it's placed on a screen wire over boiling water, the steam rising will actually help drive the primary stain into the endospores, staining them green from the malachite green. The cells will then be decolorized by water and restained by the counterstained safranin. So here you can see that these endospores were actually penetrated with the heat, and the malachite green was able to go into them, staining them green. But sometimes you can have some where you have the cell that's stained pink from the safranin, but it just contains a clear circle. And whenever you see that, it's simply because the heat wasn't able to help penetrate the primary stain into the endospores. But the circle indicates that there is an endospore there. For the flagella staining, flagella are tail-like structures that some organisms use to move from one place to another, and they're so thin that they're not visible without this staining technique. The flagella staining technique coats the flagella with stain until they're thick enough to be visualized. Although this stain has no clinical significance, visualizing flagella gives us the ability to classify and identify bacteria based on how many flagella are present and where they are on the cell body. To conduct this stain, a mordant such as tannic acid is used to flood the specimen to coat and thicken the flagella. The specimen is then stained by pyrorosmalin to stain the specimen itself. As you can see here in the image below, you can see a variety of bacteria with their flagella coming off of their cell bodies. So what did we learn about staining? Let's go over what we have learned. We learned that the staining allows us to visualize cell morphology, which is how they're shaped and arranged. It also allows us to identify unknown organisms based on the reaction of the stains done to them. We also learned that simple staining techniques utilize only one dye that either stains the cell or the background to emphasize the organism's cell morphology. And finally, we learned that differential staining techniques utilize multiple dyes to differentiate organisms into groups based on the components of their cell walls. Thank you for watching my video and taking the time. I hope that it really helps you understand staining microscopic specimens. Thank you.